Apply just a slight bit of pressure to a cooked piece of salmon, and it immediately fractures along precise repeating lines. Apply that same thermal and physical stress to a piece of beef, and the muscle shears or shreds. Aquatic animal tissue reacts to heat by breaking apart entirely intact. The mechanical cause of this clean separation is rooted entirely in the architecture of the muscle fibers. Fish muscle fibers are exceptionally short. They grow to a maximum length of only about 3 centimeters. Looking at this side-by-side comparison, mammalian tissue on the left consists of long, continuous fibers running parallel to each other. The aquatic tissue on the right is built strictly from these staggered 3-centimeter segments. To understand why a fillet behaves the way it does, we have to look at what holds these short segments together and what exactly happens to that material when heat is applied. The physical behavior and culinary texture of fish are direct mechanical results of a highly specialized aquatic morphology. This diagram of a cod fillet reveals how those short muscle segments are organized. Biologists refer to these individual blocks of muscle as myotomes. Each myotome forms a distinct undulating W-shaped segment, stacked tightly against the next. Between every W-shaped block is a narrow dividing line, labeled here as the myocomata. The myocomata are thin sheets of connective tissue. They serve as the structural framework that physically anchors the stacked myotomes in place. If we break down the composition of the muscle itself using this chart, 16 to 22% consists of sarcoplasmic proteins, such as myoalbumin and globulin. The vast majority, 75%, is made up of myofibrillar proteins, specifically actin and myosin. But the deciding factor during cooking isn't found within those muscle proteins. It lies in the specific type of collagen forming the myocomata connective sheets. Fish collagen degrades at a significantly lower temperature than the connective collagen found in land mammals. When exposed to even moderate heat, this aquatic collagen hydrolyzes completely, converting rapidly into liquid gelatin. As the myocomata liquefies, the connective framework holding the tissue together dissolves. The short, W-shaped myotone blocks are immediately freed from each other. A culinary flake of fish is simply a single, intact myotone block sliding away from the fillet after its surrounding connective tissue reaches a thermodynamic point of failure. This cross-section diagram highlights another distinct localized feature, a specialized area of dark tissue known as red muscle. Looking at the photographic insert on the right, you can see this dark band is positioned just underneath the skin, running directly alongside the fish's lateral line. This heavily pigmented dark muscle functions as a biological storehouse. It holds dense concentrations of glycogen and other metabolic energy reserves. It also maintains an extremely dense lipid profile. Up to 40% of this tissue consists of long-chain, highly unsaturated fatty acids, including phospholipids and triglycerides. The high concentration of myoglobin and energy reserves in this tissue exists entirely to fuel the locomotive demands of the animal. Demersal species, fish that feed near the ocean floor and move only periodically, require very little sustained energy. Consequently, they possess only a marginal amount of this dark muscle. This chart displays several pelagic species, such as marlin, tuna, and mackerel. These are open ocean predators that swim continuously and rarely come to rest. To sustain continuous forward propulsion in the open ocean, up to 48% of a pelagic fish's total body weight can consist entirely of this dense, dark muscle. The ratio of dark to white muscle in any given fillet acts as a structural blueprint, reflecting the exact locomotive output required by that species' evolutionary survival strategy. Looking closely at these raw cuts of salmon, the deep orange-red coloration presents a specific biochemical exception. In mammalian meat and the dark muscle of pelagic fish, a reddish hue is generated by high concentrations of myoglobin. The color of salmon and sea trout, however, is entirely unrelated to standard myoglobin oxygenation. This diagram shows the structure of astaxanthin, the specific red carotenoid responsible for the tissue's heavy coloration. Myoglobin is an oxygen-carrying protein produced internally by the muscle itself. Astaxanthin is a compound the fish must physically accumulate through its diet and store inside its tissue. While scientific research continues, current evidence clearly indicates this heavy concentration of carotenoids operates as a powerful tissue antioxidant. The visual identity of salmon is the result of highly specific dietary accumulation, separating its biochemical profile from the standard functional biology of terrestrial and marine animals alike. We can see the complexity of the living organism in this cross-section. But once the animal dies, a new series of rapid biochemical changes begins. Aquatic species undergo rigor mortis, just as mammals do, but the biological timeline operates differently. This timeline tracks post-mortem stiffening. While mammalian rigor spans a long duration, the aquatic process is significantly shorter. In fish, complete rigor mortis sets in and resolves within a window of just 1 to 7 hours after death. Simultaneously, the tissue experiences an overall drop in its internal pH level. This decrease is driven by the metabolic formation of lactic acid within the muscle fibers. Much like the accelerated rigor period, this overall drop in pH is relatively minor when measured against standard mammalian tissue degradation. From the extremely low thermal threshold of its connective collagen to the high lipid density of its pelagic dark muscle, every structural element of a fish fillet represents a highly specific thermodynamic and biochemical system optimized perfectly for life in the water.